Hi and welcome to the first module we have on protein. Protein, a bit like carbohydrate, has a huge amount of evidence and research that underpins our sort of knowledge in the sports nutrition area. So I'm going to have to try and consolidate a lot of information pretty quickly, and it really relates to three objectives that we have. So we're going to describe some guidelines and some recommendations for protein, for general nutrition as well as maintaining sort of a healthy body weight and sports performance. We're going to discuss protein during and after exercise and outline requirements, as I said, and strategies to enhance recovery and adaptation after exercise because protein's involved in that. So the first module, we're going to discuss some protein basics for those people who haven't done a lot of basic nutrition, and we're going to talk about protein metabolism, and then in the second module we'll switch over and talk a lot about protein metabolism research and where our knowledge is at, and then finish off with some recommendations. Obviously this area is fraught with lots of information. Some of that information is really high quality, some of it's poor quality, and we've got a large commercial interest in protein. So I guess the approach that I've taken to this area is to try and provide you with what scientific peer-reviewed evidence that we have, and then anything that sort of sits outside of that is up to you to interpret as either being legitimate or potentially questionable. A reminder that with regards to energy production for exercise, whether it be high intensity exercise or aerobic exercise, protein is one substrate which is used fairly sparingly by the body. It's really only used in larger amounts when we don't have adequate amounts of carbohydrate or fat available to fuel the exercise that we're undertaking, and that's done in an aerobic sense. And usually that's in the form of the use of circulating amino acids or some amino acids that might be within the cell itself. So let's start off with some protein basics. We discussed earlier in previous modules the energy content. It's about 17 kilojoules per gram of protein. Obviously, it's very much associated with meat and dairy foods, animal-based proteins, but we also importantly have proteins found in lots of foods that are plant-based. And so understanding where protein comes from is important when we're going to assess someone's total protein intake and look at whether they're eating adequately or not. A little bit of chemistry around the structure and function of proteins. They're the only compound that we have which is nitrogenous or contains nitrogen atoms. We've got carbon and hydrogen and oxygen. And we have proteins being made up of amino acids. So amino acids, this is a generic amino acid here. All proteins, or all amino acids are the same. They have an amino group, which is where the nitrogen sits. And we have an acid group, a carboxyl acid group. And then the different amino acids differ in terms of their sort of their structure off the carbon skeleton here. So we have glycine, which has got one single hydrogen ion. Another carbon and 3 hydrogens is L-alanine. So it's this component that makes the different amino acids that we have. Now we have 20 of them, and 9 of them are what we call essential amino acids. So that means that the body can't manufacture those particular amino acids from other amino acids. So we need to have those supplied by the diet. And it's these amino acids, if you like, they're the bricks that form the structures of our body. And so we have strings of these that are put together that form many, many different compounds within the body. You may see in some of the literature that I've got on Learning at Griffith, and you may hear me talk about essential or indispensable amino acids. It's much the same thing. We have 9 of those. And we also, within that, have what we call a branched chain amino acids. So leucine, isoleucine, and valine are the branched chain amino acids, and they're all essential amino acids. But we have 9 essential amino acids here. And then the remaining 11 amino acids are what we call non-essential amino acids. So in other words, we can derive these from compounds, some of the other compounds that we have. Now, we've got amino acids, and it's these amino acids that form all of the proteins of our body. It's the architecture or how these building blocks that are put together that determines what structure or what form comes. We have simple 2 amino acids joined together, so that's what we call a dipeptide. 3 amino acids joined together, tripeptide, but most proteins in the body are polypeptides, which have a whole string of amino acids bonded together. And it's those that I want to talk about now, because these longer chain amino acids can differ considerably because of a variety of different properties. So they can differ in terms of the total number of amino acids in the string. They can differ in terms of the types of amino acids that are in the string, in terms of how many different types are there. The order of those amino acids, so where they sit relative to other amino acids, can change what is produced. And when you've got very long chains of amino acids, they tend to fold or coil, and that coiling can also change the structure and function of an amino acid. So what starts off as being a very sort of simple structure can be highly complex because of these 4 different parameters that we have that can all influence what is produced by these building blocks. So it's a pretty remarkable process that we have. And this is a diagram to show you, well, this is the individual sort of structure of an amino acid sequence. But when you put all of these things together, in reality, they form these sort of coils, and then the coils form bridges within them as they fold over. As one example, here's hemoglobin. So hemoglobin is 4 highly folded polypeptide chains. So these are the protein chains here. We have a heme, which is the non-protein portion of hemoglobin, and we've got iron in the middle. And so the amino acid sequence determines the shape of the polypeptide chain, as I mentioned. So what starts off as being very simple, as you can see, can become very, very complicated, and can result in many, many different forms, which is one of the wonderful things that protein allows us to be able to do for our body. So why we need protein? Well, it does a whole range of many, many different things for us. Nails and skin, blood. We're going to spend most of our time talking about muscles and what protein does to our muscles, but it's important that it also has roles to play in immune function, cellular health, enzymes, brain and nerves. So clearly protein is critically important in terms of maintaining function of the body in a healthy way. So we have about somewhere between 15 and 20% of our body made up of protein, the total body weight that we have. Now of that, 16 odd percent, just under half of it is in the form of skeletal muscle. So it's a large proportion of the total protein that we have, and about a quarter of it is also in the form of organs, with the remaining sort of smaller amounts being devoted to skin and blood, and then you've got immunoproteins, transport proteins, and hormones. So it's spread in a variety of different environments, but obviously we're going to be, from a sports nutrition perspective, talking specifically about skeletal muscle, as I said, in a little bit more detail. A little bit on protein digestion, because that's important to be aware of as well. Protein digestion usually starts in the stomach with some pepsin and hydrochloric acid, and then there's further digestion that occurs from enzymes that are released in the pancreas. So there's peptidases in the pancreas. So as soon as the food leaves the stomach, it's partially digested, hits the small intestine. The pancreas releases these peptidases, which break down some of these proteins into sort of smaller tri and dipeptides, with the final digestion of proteins to amino acids occurring in the small intestine. The amino acids are then absorbed into the portal blood, and then they're transported to liver, and from there they enter into the general circulation. So these amino acids are in the portal blood, hit the liver, and then get distributed out to the various cells around the body, with little dietary protein being left in feces. So if you've got someone, for instance, who's unwell and they're put on a low residue diet, they can normally have reasonable amounts of protein, because the body does a great job in digesting the bulk of it, and so very little is actually excreted. Just digestion, similar diagram, but just sort of seen more as a sort of cross-sectional view. Protein coming in, the polypeptides breaking down into tri-dipeptides. This is usually in the lumen of the small intestine, so in the sort of main part of the small intestine. And then once you get into the microvilli, we have sort of the final digestion. We have amino acids then entering the blood capillary via the portal vein. So once an amino acid enters a cell, we describe that as the amino acid pool in the cell, and it can have a variety of fates. So we've got amino acids coming from the diet, amino acids being broken down from the cell itself. We can have synthesis of non-protein nitrogen-containing compounds. Proteins can be, or amino acids can be used for ketone production. We'll talk about that in some upcoming modules, not necessarily today. Glucose production from carbon skeletons. We have energy production, as I said, if we're in need of immediate sort of energy demands, and synthesis of proteins for cell structure, immune function, and so on. As I said earlier, we've got nitrogen in the amino group, and so if we've got an excess via metabolic pathways, we can produce ammonia, which gets synthesized to urea in the liver and then excreted in the kidneys. So it says amino acids can be sacrificed to provide energy if needed. Adequate intake of carbohydrate and fat spares this, and that more protein, if you're consuming more protein than you need, we do have the capability of breaking down the components of amino acids, and then some of those components can be stored as body fat. Okay, going back to our sources of protein, now that we know what proteins are and how they get digested. As I said earlier, most people recognize that meat products contain protein. In this case, we also have, that includes fish, eggs, dairy products, and then we've got these sort of plant-based proteins that we find in bread products, nuts and seeds, and in some vegetable products, such as soy products, and various sort of seeds and beans. In terms of plant proteins, I'll just blow this up so you can see it a little bit clearer. We have various sources of plant proteins, from nuts, potatoes, legumes such as chickpeas, young soybeans, edamame beans, quinoa, black beans. I think the key for this image here is to just pay attention to the serve size, because whilst these things may contain protein, the various serve sizes might be more or less easily achieved. A whole potato, for instance, you might be able to consume that. Certainly if you're having a large meal, you might consume a whole potato, which is going to give you about 8 grams of protein. In terms of things like almonds, it's about half a cup, gives you 15 grams. So they're actually very rich in protein, but you need to have a reasonably large serve of them. And when you're coming down to things like chia seeds, 3 tablespoons gives you 6 to 9 grams. So it is important to be aware of the serve size that we have and the extent to which the protein can be yielded from these products. Now, it's not just total amount of protein that's available. It's also to do with the quality of the protein in terms of whether your body can actually sort of use it and digest it and how functional it is. And so we consider primarily 2 things when looking at protein. Apart from the dose of protein, we look at the quality of the protein, which has been done a couple of different ways in terms of understanding protein quality, and to a lesser extent, the speed of the appearance of the amino acids. So one of the issues with protein quality has to do with its digestibility. And I mentioned earlier that most protein, the vast majority of it gets digested and absorbed, and that's certainly the case with animal proteins, where you get a large proportion is absorbed. But to some degree less, plant proteins are less well absorbed. Still predominantly digested and brought in, but to slightly lesser degree than what we've seen with animal products. And the other, element with comparing meat products with plant-based proteins is the indispensable amino acids, or the essential amino acids that they have. And this table that I have for you here is what we call the DIAAS, which stands for the digestible indispensable amino acid score. And so this is a way of scoring different protein sources and comparing them based on their digestibility and also their essential amino acids, or how many of these sort of critically important amino acids exist. So this table here, this is milk protein concentrate, this is whey protein isolate, this is soy protein isolate, and a whole range of other sort of whole foods here. And you can see this value here is a previous protein score that was used, but more commonly now we use this DIAAS score. And you can see here, for instance, milk, whole milk at about 1.44 is one of the sort of highest scoring protein sources. You compare that to whey protein isolate, which is 1.09, very high as well. But we have scores for things like chickpeas and tofu that are substantially lower than what we see in some of the animal-based proteins. And then on the far right here, we have what we call the limiting amino acid. So this is the amino acid that's in its lowest form, but is essential in that product. So in other words, what is the amino acid that's going to limit the production of proteins from this source? So it's good to have some understanding of different amino acid scores. And as I said, this paper is available on the Learning at Griffith site for you to have a look at and get a sense of the different amino acid scores from both plant and animal sources, as well as some supplement isolated sources. Historically, this is, I guess, how we would encourage individuals who are vegetarian to consume foods in order to ensure that we had a mix of essential amino acids when using plant-based proteins. So by combining foods that contain all of the essential amino acids. So legumes consumed with grains, for instance, you would pick up all of the essential amino acids, or you'd have less of a risk of being exposed to a lack of protein being able to be synthesized because maybe methionine or tryptophan was not present, whereas if you have a combination of both of these products, you're going to get all of those essential amino acids, so that when the cell wants to make that protein, it's got all of the building blocks that it needs to. The final thing I wanted to just touch on with regards to protein quality from a diet, if you like, has to do with amino acid speed. And there is some evidence to suggest that certain products, such as whey protein, have a more rapid appearance of critical amino acids in the bloodstream than we find with casein and casein hydrolysate. So these are the two primary proteins that are in milk. Casein and whey. And so if you have a look at this middle diagram here, for instance, which is the concentration of leucine, and I'm going to talk to you more about leucine, particularly later on in this lecture and also in the next module, because leucine appears to be a critical trigger amino acid to turn on muscle protein synthesis. So if you're getting a larger increase or a larger rapid rise in leucine, that may be to some advantage in certain circumstances, and those circumstances we'll discuss a little bit later on. But you can see here that in comparison to casein, which is this darker line down the bottom here, we're getting a much faster rise in leucine from the whey protein isolate in this case. So even though they all contain essential amino acids and have a sort of similar score, the rate of appearance for this particular important amino acid is different between the two. Okay, and the last thing that I want you to just, I guess it's important to reflect on at this point is that we have a huge industry that is producing and profiting from protein supplementation. And typically this is done in the form where the protein is extracted potentially partially digested or compartmentalized, where elements of whole proteins are broken down and made into sort of more basic components and then sold in large doses. And so with any industry where there's an opportunity to make plenty of money, there's going to be a large market of people trying to push these products. And so I guess it's important to be aware of the fact that just because these products exist, that's probably more a reflection of the commercial opportunity, more so than the scientific evidence of their benefit or harm. Okay, I'm just going to give you a couple of points now about some information that you'll see over the coming slides. So I've called this one a little bit of Back to the Future because I'm telling you a little bit about what you're going to see. So what we're going to find out is that higher protein diets promote gains in muscle mass, especially when combined with resistance training. I should have put this statement the other way around. You're not going to eat your way to big muscles. You have to train your way to big muscles, and hypertrophy of muscles or changes in mitochondrial density, and we'll talk about that in a minute. You're not going to eat your way to that, but the food that you consume in and around your training may augment or supplement the training that you're doing. Without the training, you're not going to see the adaptation occur. So that's the stimulus, and then the moderator of the stimulus typically is your food intake. It may help spare muscle mass during caloric restriction, and that's important because there's a lot of people who are under caloric restriction for a variety of reasons. It may attenuate the natural loss of muscle that accompanies aging, and with an aging population, that's critically important. Here's some broad recommendations for protein intake, and you can see here straight away that the protein intake recommendations appear as a grams per kilo body weight per day. That's how we recommend protein. Now, the recommended dietary intake for adults in Australia is about 0.8, it's 0.75 grams per kilo body weight. That's what the evidence suggests the average person who's not doing large amounts of exercise should consume in order to be adequate in their protein intake. So for the sedentary man or woman, I've got here 0.8 to 1, but the RDI is about 0.75. But you can see here that all of these values for athletic individuals sit higher than the RDI. I put a line through the male here. So this is any elite endurance athlete. You're probably looking at around almost a doubling of the RDI, about 1.6. Slightly less if you're doing less endurance activity. Football, power sports, resistance athletes who are in the early phase of their training, and resistance athletes maybe in the steady state of their training. Whilst these values are recommendations, there may be individual reasons to moderate these values. And we're going to talk in more detail about what people consume by way of protein intakes. And finally, on this slide, the consumption of approximately 20 grams, or if you convert that to a body weight, is about 0.3 grams per kilo body weight of high quality protein throughout or immediately after training, particularly resistance training, enhances this protein synthetic response. So that's a key dose to remember. And I recall from asking you guys at the start of the trimester what dose you needed for protein, and many of you were already aware that it was roughly around 20 grams as a baseline recommendation. So that's good. You may need slightly more if you're older, and I'll show you some data on that. But the good news is you can elicit the same protein synthetic response. You just may need to eat a little bit more to get that response as you age, and that the timing of the intake is important for maximizing this response. One thing that I would encourage you to sort of think about is that it's not just about muscle size. Really critically, the important thing is about muscle function. And so if we've got measures of strength, if we've got measures of endurance capacity, functional capacity is ultimately what we're looking at. Obviously, if you've got larger muscles, you've got a greater likelihood of being able to produce that muscle force or the performance that you might be looking for, particularly in a strength power athlete. For most people, people without kidney disease, having a higher protein diet is unlikely to cause kidney problems in those individuals, and we'll show you some data on that as well. So let's talk about why we're interested in protein. This is a sport and exercise nutrition class. Obviously, we're interested in protein from what it does to an athletic physique. It's important to realize that if you're talking about an endurance athlete, we're often talking about mitochondrial proteins. So these are the energy producing components of the cell, and we want to produce a large number of mitochondria. We want the mitochondria to be healthy. We want the mitochondria turnover to be producing and reproducing more good quality mitochondrial proteins over a sustained period. And these proteins are inherent within the muscle. It's not necessarily associated with muscle size. So Chris Froome here, who's a Tour de France winner that you have in front of the sumo wrestlers, has extremely good protein adaptation, but it's in the form of mitochondrial proteins, not necessarily in the form of hypertrophy of the myofibrillar proteins that we see in strength and power sports. So this is really about the force producing component and the muscle size and the cross-sectional area of the muscle to be able to produce power and strength. And so you can see from an appearance perspective that these individuals, although they're both highly refined in their protein characteristics, the expression is very different protein structures, and therefore the appearance is very, very different. And we see this across sports, going from people who run a 100-meter dash right through to people who run a marathon, so strength power through to more pure endurance. But we're also, from an athletic perspective, interested in protein from an injury, a rehab, a recovery perspective. I'm not exactly sure what sort of injury this one is, but nonetheless, I thought it was an interesting example of injury and rehab. But one of the other key elements that's associated with sport and exercise nutrition that we're interested in from a protein perspective is individuals who work in very strenuous conditions, where physically they're put through very demanding paces, if you like, and they may or may not have access to a lot of protein available to support the work that they do, and that can be emergency services. There's a great deal of research that happens in protein that is associated with space, because we've got to travel long periods of time, and we're also in a weightless environment, and so the effect of the stimulus that we see from resistance type activity is much harder to construct in a weightless environment. And people in military environments as well, who may be under high amounts of stress, but with limited access to protein. We're obviously also interested in protein from a growth perspective, in terms of not just people who do sport, but if you're doing sport as you're developing and you're growing, to what extent do you need to moderate your protein intake? I mentioned elderly, and we're dealing with an older population, so understanding what happens to protein requirements as you age is critically important. And then we also have people who are unwell, who may or may not be involved in sport, but may have high levels of protein turnover. So Jonah Lomu, for those people who are unfamiliar with rugby, was an All Black. He was at one point in time by far the most dominant rugby player on the planet. And Jonah unfortunately passed away several years ago now, and had some severe kidney issues, so was undergoing dialysis as part of his sort of medical treatment. And in that environment, you've got a high need to manage and monitor protein requirements, because your kidneys are, as we saw earlier, involved in protein excretion. And so you need to manage protein intake so that the person's not breaking down their own protein and having to excrete that, and equally they're not having a high protein diet, which results in a higher need to excrete the nitrogen from the diet. Okay, so I mentioned earlier that we wanted to, we would have a look at what athletes might consume by way of protein, and I thought we'll start at the top and go and have a look at individuals who are trying to maximize skeletal hypertrophy. So individuals who are trying to challenge their own physique and aspire to grow as much muscle as possible. And so there is some literature on this. This is a really nice study that was published a few years ago now in the journal Sports Medicine, where the data that was on competitive bodybuilders and what they eat to try and understand that if you're trying to push the boundaries of hypertrophy, what are these individuals doing? And this is what this study found. It's a little bit difficult to read, but it's a really nice paper because it summarizes the number of athletes, so these are the authors of the paper, the number of athletes in the study, but it also describes the phase. So NS means that it's not specified. CP means that it's competition preparation, so they were heading into a competition. C means that they were actually in a competition, and so it gives you an indication as to what extent their food intake may vary, and some of these studies have done competition preparation and then within competition. For those people who are interested in energy, it provides the energy value in terms of kilojoules per day, but because we're talking about protein, I thought we'd summarise the protein component here. And you can see across this table, I've summarised it here, for the males, the protein intakes of these competitive bodybuilders range from 157 to 406 grams of protein per day. So in relative terms, that's 1.9 to 4.3 grams per kilo body weight. Now, I've never seen a study that suggests that you need to consume this upper limit of a protein intake here. But 1.9 is certainly close to the recommendation that I had in my earlier table, and would not be considered extremely excessive when you consider this population of individuals. The other good thing about this paper is it also has female bodybuilders, and we have our values here, similar, our protein values, as low as 48 grams a day up to 162 grams a day. So that actually equated in terms of per kilo body weight of these individuals to effectively the RDI at 0.8 grams up to 2.8 grams. So again, we've got a wide variety of individuals in terms of their dietary intake, even though they are all trying to maximize hypertrophy. So what that says to me is you probably need to eat, in what this is telling us is that the anecdotal evidence or the lived belief of these individuals is that you need to eat more than is recommended in terms of protein, but you can achieve hypertrophy from a range of protein intakes, and there's probably an upper ceiling of that, at which point eating more, you're going to get a diminishing return on that investment. Now, it's not obviously just protein intake. You need to be in what we call an energy surplus or a positive energy balance, which is why this study also has the energy values there. And no doubt, your protein intake and how important that is will be somewhat determined by how much energy you've got. And so if you've got a relatively low energy intake, then having a higher protein intake might be more critical in that instance, and vice versa. The next image that I wanted to show you came from a fairly recent paper that I thought had a great diagram in it to help us understand the factors or the elements at play here. If you want to do some deep reading of understanding how the body regulates hypertrophy, this paper I've put it as extended reading on the Learning at Griffith site. It's by Lim, is the first author. And it goes into a considerable amount of detail to understand these internal variables that regulate how your body responds to the external variables that may stimulate or moderate hypertrophy in an individual. So things like mechanotransduction, the mTOR signaling pathway, so that's mammalian target of rapamycin signaling pathway, ribosomal biogenesis, and a range of other components. All of these things we can't control. They're all internal to our genetic sort of makeup and relate to our signaling pathways that we have. But we have these external variables here, and RE is resistance exercise. The green one here is diet, then we have sleep, and then we have supplements. And the thing that I like about this is it gives you an indication of the scale of the importance of these things, if ultimately what we're looking for is hypertrophy as our output. And so our internal variables are critically important. You have to have, it's far easier to have muscle hypertrophy if you've got the genetic makeup, these internal components that facilitate hypertrophy. And then from there, it's the resistance exercise, which is the stimulus, and then a dietary component, a sleep component, and much, much smaller is the supplement component. So I really like the way that this diagram unpacked how do you grow larger muscles, and what are the inputs, and what are the things you can control. And if you want to read more about how the body regulates this, this is a great paper that unpacks all of that. What I want to do now briefly is just talk about skeletal muscle turnover and how we monitor it in research. Historically, we've used indirect methods. So you may have heard of nitrogen balance, which is a whole body model for protein regulation, where we look at total protein turnover in a day. But we have a number of direct methods, and these involve looking at sort of molecular signaling. So what's happening at the molecular level, which genes are being turned on, which proteins are being sort of upregulated, and can we monitor that in response to different training, and that might give us an indication as to which training or which dietary strategy is giving the strongest signal, if you like. And then we have these other direct measures where we use amino acid traces, where we're actually looking at amino acids that are coming into a muscle cell and maybe being taken up as a whole protein or being left as an amino acid. So one of these, the protein signaling, is really around molecular biology, and the second one is more about a metabolic physiology. The indirect measure, the nitrogen balance measure, this gives you a sort of an overall intake of what's done here. So if you have a protein intake, and in this case the example is a 90-gram protein intake, it enters your gut. Some of that protein is absorbed. We have proteins entering the gut from the liver, so we mentioned sort of digestive enzymes, other amino acids that sit in the gut, and so you've got about 70 grams entering the gut from sort of liver sort of circulation. Some of this also helps to perform digestive processes and binding of fats and other components, and you end up with this 160 odd grams coming from these two sources, and you absorb about 150 grams of that 160, leaving you with a 10 gram fecal excretion heading out in this direction. So we've got this sort of cycling of amino acids coming in. They come into the liver. The liver, as we said, produces some urinary nitrogen, and then these other inputs then head into the body and can be used, as we said, for muscle, organs, plasma proteins, and a whole range of different things. And here's some values here for overall protein synthesis in the day. So if we were to monitor dietary intake, and we know how much protein's coming in, and we're monitoring outputs, so fecal outputs and urinary outputs, we can then determine what is the overall circulation, if you like, of amino acids that goes into this area over here. Now, the challenge with this is that we don't know where these amino acids are going and what they're doing, but we do have a feel for how much comes in, how much goes out, and if we collect urine, we know how much is exited this pathway, so we can, by deduction, work out what's circulating in here. And for an 80 kilo man, it's sort of typically estimated that you get about 250 grams of protein synthesis per day. So there's 250 grams of new protein being generated every day across all of the forms of protein that we have. And in terms of actual weight, that would mean that you would replace all of the proteins of your body, or be in a completely sort of new body, if you like, in just under a year, which is great news. If you like the idea of being a new you, we are effectively a new us, usually in just under a year, which is provided that you've got an adequate protein intake, which is pretty fascinating to think about. Got myself a low battery here. I'll push on for a second because we're not too far away. So we've got this direct signaling pathway, and it is very complicated. And because it's so complicated, I don't expect you to know too much about it, other than to be aware that we can monitor these protein signals that come as a response of the training that we're doing. And so, sorry, just plugging in. We have, I mentioned before, mTOR, the mammalian target of rapamycin, is one of the sort of central controllers here, but we have amino acid signals, insulin-like growth factors, having an influence on what signals are occurring. The ATP status of the muscle cell is also sending signals, and we can monitor many of these pathways here to work out what's being upregulated. Now, just to highlight how complicated this is the upstream regulators of the mTORC1 signaling pathway, and so it becomes far more challenging. We have amino acids up here. We have inflammation and a whole range of other sort of stimulus. And there's a really nice comprehensive review of the mTORC signaling pathway for the body. And this is what it looks like here. So we've got the mTORC1 here, we've got mTORC2 over here. And this is one of the reasons that we won't be going into it in this course, is because it's well beyond my own understanding, and really involves heavy molecular biology. One thing that I would encourage you to think about, though, when looking at signaling type studies, is that because of this complexity in nature, when we've got things that are complex, multi-layered, it is often associated with a certain element of redundance. So what I mean by that is you may see a signal, but that signal may not necessarily translate into something that's important by way of a sort of functional change. And here's one example of a recent paper that talks about myofibrillar protein synthesis. So muscle cell synthesis rates are increased in chronically exercised skeletal muscle despite a decrease in anabolic signaling. So even though we're getting less of the signal, we're still getting good responses, which is what we're looking for from the actual muscle itself. So it's not as simple as saying signal equals output. Signal may be important in certain phases. Some signals may be redundant and actually it doesn't result in a functional change, and others clearly do. So it is an area to be aware of the complexity, but also be aware of the limitations of seeing some of the outcomes in some of these studies. Nonetheless, fascinating. I do want to talk more about direct measures. And so if we talk about amino acids measured directly, this schematic here is something that I've sort of put together to try and help you understand what is being monitored in these studies. So if we think about a muscle cell, this is a muscle cell above the line here, and this is blood. So we've got circulating amino acids, and some of these amino acids come in and some of these amino acids coming out. So this is the cell's amino acid pool. So we've got this amino acid sitting here, and now some of those amino acids are being converted up into muscle protein, and muscle proteins being regenerated all the time and broken down all the time. So we have this synthesis component, we have a breakdown component. And we have an oxidation component as well. So some of these amino acids are being transaminated and going and being oxidized. What we do by way of these particular studies is usually provide some amino acid that's labeled. It's got a tracer on it in the bloodstream. And so we know how much of a particular amino acid is coming in, and we can also monitor how much of that amino acid is going out on the other side of the muscle. And from that, we can then determine how much of this amino acid has come into the muscle cell. So how much has been absorbed into the muscle cell. Now, if we also do a muscle biopsy of the muscle itself, that can then tell us how much of the labeled amino acid, which is in here, has been converted up into a muscle protein. And from that, we can work out not an indirect, but a direct measure of how much amino acid is being utilized and then incorporated up into muscle protein. And so we have studies that can give us an indication of synthesis as well as potential breakdown. Now, from a methodological perspective, it's far easier and far cheaper to measure synthesis. So you'll see a lot of the studies only measure muscle protein synthetic response. There is another method, which is known as the indispensable amino acid oxidation method. This is a cheaper, less complicated method, which looks at oxidation of amino acids. So basically the way this works is that it works under the assumption that when one essential amino acid is deficient for synthesis, then all of the other amino acids, including an indicator amino acid, which is usually phenylalanine, will be oxidized. So the way these studies work in terms of determining amino acid need is to look at, in this case, this is phenylalanine, it's labeled again, and they're giving lysine. And so we have high levels of oxidation of phenylalanine here when we've got very low intakes of lysine. So in other words, some of the lysine is being used to make proteins, but there's a lot of phenylalanine there that is not able to be to be used as protein because we've got an insufficient intake of lysine, so the body just gets rid of it. Now, as lysine intake increases, and what we have here is an oral dose or an IV infusion here. This was a study that was looking at, can we just give an oral dose versus an IV infusion? What you see is as the dose increases, we see a reduction in the amount of phenylalanine that's being oxidized. So it's being incorporated into proteins. And you get to a point where it's like a break point. So in other words, at this point here, we know that the lysine is at 40 grams, or 40 milligrams per kilogram per day in this case, is then able to incorporate as much phenylalanine as it can, and we end up with this break point or this flat line. We're having more lysine is not producing more phenylalanine because this is still just being incorporated into protein. So this amino acid indicator method just relies on supply of a labeled amino acid and an intake, and then monitoring. In this case, we can measure this in the breath, this CO2 phenylalanine concentration. So there's no muscle biopsies involved, and so therefore it's a less invasive technique. And it's a little bit sort of newer than the tracer technique. So there's a whole large amount of technical requirement to look at the influence of nutrition and exercise using these direct tracer methods. And there's only a few labs in the world that do it. And so here's some of the people who are involved in some of that work, because I think it's important to understand the background or the people who sort of sit behind this work. Now, if we have a look at protein balance across a day, we might be not bringing in protein, and at this point we're in what we might call a negative sort of protein balance, so we're sort of breaking components down. Then we have a meal and we get some amino acids, and we're starting to grow, have a synthesis of protein, another meal and so on. Bless me. And so if we were to take a supplement in the afternoon, here the idea would be that you're actually producing a greater opportunity to turn a deficit into a positive, and ultimately be synthesizing more amino acids. But of course, when you take a supplement, there can be a variety of responses. So if we have a meal and we get protein synthesis from that meal, you could take a supplement and you get a direct significant benefit immediately, which is additional from the supplement itself. It could be that the supplement, provides a small benefit, but not to the extent that you'd expect based on the dose of the supplement, because the body's capability of upregulating protein synthesis is not that great. So maybe you oxidize some of those amino acids. You might get a small benefit, but it's not the extent that you might think. Or alternatively,
the supplement then starts to offset some of the meal in terms of the amino acids that were available in the meal, and maybe you're now oxidizing some of those because you've got a supplement that's being produced. So obviously for most circumstances, most individuals are interested in growing muscle, whether that be hypertrophy or, as I said, mitochondrial biogenesis. Either way, we want more synthesis than breakdown, and for that to be healthy synthesis. So in this case here, if we're in a situation where our ability is to either increase synthesis or decrease breakdown, that puts the balance in favor of what we're interested in. And the second module, we're going to unpack this and look at strategies to increase muscle synthesis relative to breakdown. So thanks very much.